Hello, folks. This is Tolbert's biology class, and this is week 3. This is the lesson for week 3, and this is going to be some notes. So we're just basically going to take notes from these here slides. I have included inside your Schoology folder the actual PowerPoint. So if you need the PowerPoint, it's there. I've also included every single video that's on this particular presentation. I will refer to them in the presentation, but if you'd like to see them in their entirety, you'll have to go to your little, inside the Schoology, inside the little folder, there's a one that tells you where the videos are. So here's the knowledge that we're going to be getting today. So just as also just a little point of reference, read words and phrases are important for the assignment that you're going to be doing in this DNA replication part of our assignment in this week 3 in biology. Before a cell divides, its DNA must first be copied. How might the double helix structure of DNA make that possible? DNA crash course video. So this video, it's about 12 minutes. So if you'd like to see that DNA crash course, it's inside of the folder inside of Schoology. What role does DNA polymerase play in copying DNA? Here's the answer to that. DNA polymerase is an enzyme that joins individual nucleotides to produce a new strand of DNA. It's red words. These red words you should be writing down in your guided notes. These are the things that are important for you to have. Base pairing in a double helix explained how DNA could be copied or replicated because each base has one strand pairs with only one base and the opposite strand. Each strand of the double helix has all the information needed to reconstruct the other half by the mechanism of base pairing. Each strand, because each strand can be used to make the other strand, the strands are said to be complementary. Before a cell divides, it duplicates its DNA in the copying process called replication. So when the DNA makes a copy of its DNA, that process is called replication. This process ensures that each resulting cell has the same complete set of DNA molecules. During replication, the DNA molecule separates into two strands and then produces two new complementary strands following the rules of base pairing. Each strand of the double helix of DNA serves as a template or model for the new strand. And you can see it down here at the bottom. You have the DNA here that's all together, and after it gets unzipped, you'll have two complementary strands that we can use to make copies of the DNA. The two strands of the double helix separate or unzip, allowing two replication forks to form. The molecule that does that is called heliocase or helicase. It unzips your genes. So there is a special type of little molecule here that comes in on that's wound up, and it comes here and it basically breaks this apart. That's where the helicase comes in and does its work. It unzips your genes. As each new strand forms, new bases are added following the rules of base pairing. If the base on the old strand is adenine, then thymine is added on the newly formed strand. Likewise, guanine always paired with cytosine. The result of replication is two DNA molecules identical to each other and to the original molecule. Each DNA molecule resulting from replication has one original strand and one new strand of DNA. Also, folks, I want you to notice that once they start making this replication process, it's going to be going in several directions showed on this particular picture. So notice the picture. And also the picture is showing you our bases, being adenine, thymine, cytosine, and guanine. DNA replication is carried out by a series of enzymes. They first unzip the molecule of DNA by breaking the hydrogen bonds between base pairs and unwinding the two strands of the molecule. Each strand then serves as a template for the attachment of the complementary bases. The principal enzyme involved in DNA replication is DNA polymerase. DNA polymerase. DNA polymerase is an enzyme that joins individual nucleotides to produce a new strand of DNA. DNA polymerase also proofreads each DNA strand, ensuring that each molecule is a perfect copy of original. This is probably one of the most important steps because if there's a mistake, that's where the mistake is going to be at. So it has to be proofread. So lots of our little genetic diseases, sometimes it's in the proofreading that the diseases take hold. This DNA polymerase is able to say, okay, it's supposed to be A to the T, C to the G, and if there's an issue, then different letters are there, and you're looking at an entirely different sequence. Telomeres. Telomeres. There's two videos on telomeres. One of the videos is what is a telomere? The other one is just more on telomeres. The tips of the chromosomes are known as telomeres. The ends of DNA molecules located at the telomeres are particularly difficult to copy. Over time, DNA actually can be lost from the telomeres each time the chromosome is replicated. An enzyme called telomerase compensates for this problem by adding a short little repeated DNA sequence to the telomeres, lengthening the chromosome slightly and making it less likely to be an important gene sequence that will be lost from the telomeres during replication. So if the ends of your chromosomes, if the ends of those get shorter and shorter and there's no information there, that there's vital information that is no longer being reproduced, or there could be something your body is no longer doing if the cells are no longer giving it the instructions to do that. That's where the telomeres are, that there's a chromosome, and at the very end of it are what we call the telomeres. Okay, there is a video that's attached here. It's a quick review. Also, the review will do all the replication, but I believe you need Flash if you want to watch that particular review. How does DNA replication differ in prokaryotic cells than eukaryotic cells? Replication in most prokaryotic cells starts at a single point and proceeds in two directions until the entire chromosome is copied. In eukaryotic cells, replication may begin in dozens and even hundreds of different places on the DNA molecule, proceeding in both directions until the entire chromosome is copied. This is also important, folks, because remember, in eukaryotic cells, there's probably 1,000 times as much information that's going to need to be copied. So it's nice that we are going to be able to start that copying process in hundreds of different places. The cells of most prokaryotes have a single circular DNA molecule in the cytoplasm, containing nearly all the cell's genetic information. Eukaryotic cells, on the other hand, can have up to 1,000 times more DNA. Nearly all of the DNA of eukaryotic cells is found in the nucleus. In most prokaryotes, DNA replication does not start until the regulatory proteins bind to a single starting point on the chromosome. This triggers the beginning of DNA replication, and it goes in both directions around the circle until it's copied. Replication in most prokaryotic cells starts at a single point and proceeds in two directions until the entire chromosome is copied. Often, two chromosomes produced by replication are attached at different points inside the cell membrane and are separated when the cell splits to form two new cells. In our cells, our eukaryotic chromosomes are generally much bigger than those of prokaryotic cells. In eukaryotic cells, replication may begin at dozens or hundreds of places on the DNA molecule, proceeding in both directions until each chromosome is completely copied. Two copies of DNA produced by replication in each chromosome remain closely associated until it enters prophase of mitosis. At that point, the chromosomes condense and the two chromatids in each chromosome become clearly visible. They separate from each other in anaphase of mitosis, producing two cells, each with a complete set of genes coded in DNA. Here are the two other movies. These other two movies are linked inside of your Schoology one. So one is just basically DNA. It goes over all of that replication things that we just covered in our slides. And of course, I always like to have a DNA song. So there's a DNA song that's on there that we'll be talking about replication. Hope you're having a good week. This is part one. This is getting the knowledge, and taking notes is part of getting the knowledge. The next step you should be moving to is the application of the knowledge.